This video is about the G1 decisions. There are two specialized non-dividing states that relate to the cell cycle, which are discussed here. The first that I'll go through is the G0 phase, and this is a period of withdrawal from the cell cycle. This is not from our textbook, and basically you can see the cell cycle, which we've talked about quite a bit here. As we are in G1, the cell sometimes can kind of take a little side step and hang out in a phase called G0, which I will write some notes about now. So here's some information about G0. G0 is prolonged, but it is not permanent. Cells can sense growth factors, and if so, they can move back into G1, which we would see here. During G0, cells are not actively making cyclins or any other of the protein prep that they might use to move through G1. And one example of cells that are in G0 are certain liver cells. They actually divide about once per year, and these cells are stuck in G0 until they get ready to divide. This is very slow. Frequency of cell division for these certain specialized liver cells compared to some other cells we've talked about. For example, the intestinal epithelial cells, which on average divide about twice per day. Now let's talk about the other specialized non-dividing state that relates to the cell cycle. This is terminal differentiation. This is a permanent withdrawal from the cell cycle and includes dismantling cell cycle control systems. Some cells that do this include neurons and muscle cells. One way to think of this, which is pretty interesting, is to note that brain cancers, as far as brain cancers from neurons, are incredibly rare as compared to other tissues because the neurons are not even dividing. One exception is glial cells. Glial cells are not neurons, but they are part of the brain. And glial cells are not terminally differentiated, and they can produce brain cancers, which are called gliomas. Now we're going to talk about options for a cell, which is in G1. And so this is a kind of cool figure from your textbook, and kind of unusual compared to what we usually see as far as like the perspective of proteins, but here we're looking at the perspective of a cell. And so the cell that's in G1 is right here. And basically it's got several different options about different directions that it can go. And so what we want to understand is what those options are, and then what sort of cues factor into the decision of the cell for which of those options it takes. So as a reminder, G1 is a period of metabolic activity. It's also a time of cell growth. And as we see here, the cell that's in G1 is actually migrating. So we can see like the leading edge, and we can see lamellipodia here. And most of our cells are in the G1 phase. Remember, that's the longest phase of the cell cycle, and that is a really common spot, kind of a cell cycle point for cells to be in. And here are the decision factors that a cell can take into consideration, which will determine what happens next. So one of the decision factors includes this category of extrinsic cues, extrinsic like external, and the big one is growth factors, or GFs. These are also known as mitogens. I can't hit hard enough just how important growth factors are. They're going to show up in so many of these problems, and I'm going to ask you this question multiple times about how a cell might be able to respond to growth factors and when, and that is during G1, so G1 is the only time when the cell can respond to growth factors. A second category of decision factors for a cell that will determine which of the options it takes from G1 include the concentration of cyclins, CDK activity, and of course those are tied together, and also DNA damage. And again, intrinsic cues implies internal cues for the cell, things that are inside the cell membrane. Versus extrinsic is talking about outside the cell, and so not outside the whole body of the organism, but just outside the cell. Okay, so here are the options. Basically there are 4 of them, and remember that we're starting off with our cell that's right here. So the first option is that the cell can transiently hold in G1. We call this G1 arrest. And so that's symbolized by this little volcano-looking thing here, which it looks like the cell is just kind of like in a holding pattern, kind of vibrating there with these little things here. So it's just hanging out, and this is in what is considered G1 arrest. A second option is that the cell could go in a different direction For example, this isn't saying migrating, just saying as far as the proteins that are present and what the cell is able or is unable to do. A second option is that the cell that's in G1 could go over here into G0. So this is the second option, and this is, as a reminder, a prolonged state, and the cells are not proliferating or dividing, they're not actively dividing, but they have not completely dismantled all their machinery to do so. They're just kind of like at a rest area here, and I think this is kind of cute that your textbook put a little picnic table there. A third option for a cell that's in G1 is that it might terminally differentiate. So that's option 3 over here. And so the cell has now become what looks like this kind of creepy looking like hand thing. It's very three-dimensional. So this is very special shape and implies that this cell is committed to doing essentially its job, kind of settling down, not ever going back into a process of mitosis again. And so some cells do this within a tissue or within the body. And a fourth option for the cell is shown up here. So this cell that was in G1 can move through this checkpoint, which we see here, and notice it's called the G1 checkpoint. And then this cell looks like, woo, it's really moving fast. It's not actually going anywhere again, but probably there's a lot of different proteins that are being made, cascade of gene expression that will set up the cell to start to replicate its DNA. And so this is a really crucial decision point, and it's kind of a point of no return in many ways for this cell, at least as it enters at the S phase. And I just wanted to add one quick label here to remind you that the cell that's hanging out right here, this is the cell that's in G1 arrest. This sort of implies that the cell has to go through G1 arrest to go into these other phases. I wouldn't say that's exactly true, but I think it's just showing kind of the cell that's hanging out at the crossroads here. And so if it hangs out at the crossroads, if you will, then it's in G1 arrest. But a cell that's in G1 could receive certain cues and go right into the S phase, or it could go hang out in G0, or it could go through terminal differentiation. And I want to talk a little bit more about this decision about proceeding into S phase. So moving from G1 to S is called start in yeast cells, signifying what a start of the cell cycle and basically start of this commitment to move forward with cell division. Though certainly there are a couple other checkpoints afterwards, but this is like really the biggest checkpoint. And the big concept is that after passing the G1 to S decision point, the cell cycle moves rapidly. And as a reminder, there are actually 2 checkpoints that are back-to-back within G1 to S. So these are checkpoints. And this includes the R point, which is also known as the restriction point. This seems a little weird, but I think as you go through the worksheet, you'll see a little bit more about the distinction between the things that are happening in G1 to S versus R point. It's essentially one really big signaling pathway, and so scientists have broken it down into one big checkpoint and then a second one. Though really they are essentially can be back-to-back if the proper cues are present. So let's talk about some more details about how a cell is stably inactivated in G1. As I mentioned, many of the cells of our body are in G1, and basically somatic cells of an adult spend a lot of time in G1, so this requires CDKs to be inactive. So I want to talk about what determines CDK activity state. So as a reminder, CDK activity is primarily determined by the concentration of cyclin, and that's a balance between cyclin production and cyclin degradation. What you're going to see with the worksheet is that there's a retinoblastoma or RB signaling pathway, which can culminate in the cues for transcription of certain cyclins. And so this, of course, would be necessary for cyclin protein production, and so this is a big regulation point. So cyclin concentration is a key factor for CDK activity. There's a second factor that's part of this system that slows down the cell cycle, and which I have called the negative regulation and the brake, and that's the CDKIs. So the CDKIs that will bind to and inhibit the cyclin CDK combo. And together, these 2 aspects of regulation can lead to a stable G1 phase, and the cell cannot enter the rapid cycling. So next slide, we're going to talk a little bit about this RB or retinoblastoma situation. So there is a graphic image on the next slide, just as a warning. And that image relates to an eye cancer, which is what the retinoblastoma signaling pathway and protein are named for. So we're going to talk a little bit about this childhood cancer, which is called retinoblastoma. And retino, of course, refers to retina, so the eye, and blastoma has to do with a set of cells, extra cells in this case. It's a type of cancer. A key protein that has been determined to be involved with this retinoblastoma has been named RB, and it normally functions as a tumor suppressor, which means that it would normally be functioning as a brake in the cell cycle, slowing that down. And so mutations, especially loss of function mutations in RB or retinoblastoma, would then speed up the cell cycle. And if we look a little bit at the eye structure, this is not an anatomy course at all, but this would be a healthy eye. And so here's for example, a type of tumor, the retinoblastoma, and where that would be growing within the eye. The images here show children that have retinoblastoma and some various degrees of the retinoblastoma, and we see some really advanced cases, for example down here where the eye is very impacted by this growth of cells. And so as it turns out, so one of the reasons why we know so much about this is that this can be detected, and it is common in children. Basically, if people are born with one hit or one mutation in their retinoblastoma gene, it's highly likely that within their childhood that they will arise another mutation within those cells. So you don't need to know a whole lot about that, but this is an example of a particular cancer that is just very likely to happen in children. This should sound familiar, but it is a key take-home message for the course, so I'm going to run through it real quickly again. Remember that G1 phase is primarily determined by the presence of mitogens, which are mitosis-generating molecules, mitogen, also known as growth factors. So mitogens are these extracellular ligands that can trigger cell division, in other words, progression through the cell cycle, which includes mitosis or meiosis. And so mitogens can arise either from paracrine signaling from nearby cells that secrete growth factors, or they could be from endocrine long-distance signaling when cells are far away, and then growth factors would move through the bloodstream. As a quick flashback to this cell signaling pathway, so growth factor, the extracellular ligand, is shown here in red. This is our receptor tyrosine kinase that's activated. We're going to ignore these steps right now, though they're still happening. And then what we're going to pick up with is to talk about the MAP kinase cascade. So you definitely need to know this first part about growth factor and RTKs, and then we're going to skip these parts, go to MAP kinase cascade. And more specifically, we're going to talk about here, and this is what you'll see for the detailed RB signaling pathway, that some of the targets of the MAP kinase cascade include cyclins, and MAP kinase cascade also will disable CDKIs. So on the worksheet, you'll see this pathway. You're not responsible for memorizing it, however, you should be able to work with it. And basically, one thing that I want to bring your attention to is that I messed up one label here. So this little orange guy is labeled EF2 on some of the documents, if not all of them, and over here as well. And it's actually E2F. So either way that you use it is fine with me. Obviously, I couldn't keep it straight, and just basically watch out for that. And what I really hope that you can see through this is how we're going to start off with growth factors, work with receptor tyrosine kinases, go through the MAP kinase cascade. We've got another molecule here that we haven't seen before, MYC, which I'm going to keep your eye on, things like the location where the signaling is happening, cyclins, CDKs, CDKIs. This is another part of the worksheet that you'll see, and basically we're starting off looking at DNA damage here at the top. And so basically I want you to look through this signaling pathway, so I'm not going to talk about it a whole lot, but I will give you a little kind of summary of that on the next slide. So this is some of this is in addition to what you saw on the last slide, and basically here what we're doing is checking out how DNA damage can affect the cell cycle. So DNA damage can be due from X-rays. It can also be due from chemical mutagens and other types of radiation. And so basically this can activate a kinase, which is known as ATM kinase, FYI. And then here we can see the protein that is activated and then causes a cascade of events which tie into some of our cell cycle components which we've seen so far. And as a reminder, TXN is my abbreviation for transcription, TLN is my abbreviation for translation.